Okay, this is so cool. So what happens when you cut this slime mold, Physarum polycephalum, this bright yellow thing here? This is all one single cell, and for most cells that are microscopic, if you puncture them or cut them or something like that, one hit KO. They're not going to recover from something like that usually. But this is obviously a strange case. It's spread itself very thin in some of these places, and it has this circulatory system. You see the veins here, where it's actively pumping cellular material around. This is very clearly a strange organism, right? So what happens when you cut it? Now, if you just sever one of these veins, it will just repair that, and that takes a matter of like hours. It's very fast. But if you actually pluck one of these oats, this is what it's sitting on, oats, that's what it eats, one of the things that it eats. If you pluck one of these oats out, covered in its little layer of slime, pluck it out, put it into a new dish, that's subculturing. That's a routine lab procedure. That's how we split the culture and grow more of them, and that's exactly what will happen. That oat flake will then grow as an independent specimen, and both of them will heal. It's actually like a minor cut to them when you sever these. They have like this clotting. it's like cytosol, the cytoplasm that has this like clotting feature where it'll close back its membrane. It's fascinating. So cool. But it will. Both specimens will repair themselves, and the smaller specimen that you transferred will grow normally and expand. But this actually becomes a bit of a problem when you have these two cultures, because this is an acellular slime mold. And I'll probably make another video about what exactly that means, but it prefers to be aggregated in this state with just an enormous cell with millions of nuclei, and this is its default. And if it meets another specimen in the wild that is the same species, they will fuse. In fact, it's not just if they happen upon each other. They actively send out aggressive chemical signaling to essentially call to any other specimens in the area to converge on a single point and then to fuse. And because this is the way that they can survive the very best, is they join together and unify into just the largest single cell that they can manage. And this becomes a problem in the lab, because if you split them, they're both specimens are still sending out this very powerful chemical signal to attract each other. And this happens. The specimen just got very large, obviously, and started to escape. These are just master escape artists. They can fit their whole cell through just a tiny pinprick hole by gradually pumping it through. Just one vein goes straight through the hole, pumps it through. Obviously, that's not what it did here. It was able to just crawl over the lip of the vent of the petri dish and escape very effectively. But it was calling to another specimen that we had over here, and let me show you. And this happens. So we just had these petri dishes stacked on top of each other. The cultures overgrew. They were all calling to each other with this chemical signal, so they all knew that each specimen was there, and they just found the fastest route to escape their petri dishes and all fuse into this one enormous specimen, taking up multiple petri dishes. It keeps on going too. The stack got taller and just taking up many petri dishes. And yes, so they're kind of there's a lot of ways around this, obviously, but that's a learning curve, figuring out how to culture multiple specimens in your incubator at one time, because they're constantly calling each other with this chemical signal to group back up and unify, which is so cool.